Hi guys, welcome back to another Scientific Weekly. Today we are going to talk about storage within DNA. So we have multiple sources today. We have SciNews, NowGeeks and ResearchGate. So storage in DNA, it's been theorized in the 1960s by Richard Feynman, who actually was a pretty good known physicist. He was among the top of the physicists back then, but he actually envisioned this idea and now we are making it a real thing. So storage in DNA, bacterial DNA, is not something new, but the thing that is new here is the fact that they did it in living bacteria, on the opposite of dead bacteria cells. Before they got up to roughly 200 MBs of digital storage worth in a dead cell, but now they have been able to put some useful information in E. coli in a living E. coli cell. So E. coli, nothing bad, nothing dangerous as long as it remains in the petri dish and as long as you don't eat it. Our body contains E. coli themselves, so that helps a bit. It's not deadly. Anyway, since we have multiple sources, I try to scale down most of the information a bit into one page in my notebook. And let's put the facts in a row for a moment. So this is the first time it has been done in living bacteria. So they managed to use the typical CRISPR-Cas9 method, which is pretty common in genetic modification idea. They use that to put in new pieces of DNA in the bacteria, and they could just function and replicate further without issue. So it's very small, if you can imagine, and it's the first time in bacterial in living microbes, which I already said multiple times now. So in a single gram of DNA you can get roughly 215 gigabyte. Am I saying that right? I'm not saying that right. 250 million gigabytes in 1 gram of bacterial DNA. One single gram of DNA contains much more storage than my computer system, times 10. And I also use my computer system for photography. And that's quite a lot, because I'm running out of space already. So yes, that's a lot. So it actually was done by Dr. Seth Shipman, a postdoctoral fellow at Harvard Medical School and author of the paper. Look, I missed that link before. Nature.com, we got 4 sources. But once again, Nature.com, you have to pay to access the whole article. Which is a shame. But anyway, they managed to do it. They did it with a 99% accuracy. The image here, the GIF you see is left is the original, right is the reconstructed image from the bacteria. You can see that the detail is very high and crisp for the little information they actually have. And the error in flaws is pretty minimal. There are some dots around, yes, but it looks good, it works, it's not annoying, so it's a good start. Did we have any more useful information around? Because I wrote nothing else down. Yes, beside the GIF, they also started with the image from our hand, and it looks stunningly amazing. Compare the original to the reconstructed image and you see it's basically the same. It's the same image, it has the same outcome. So the potential is there, the possibility is there, and we have the technology to do it. So why aren't we doing this? Well, because it's bloody hard. Right, I said that CRISPR-Cas is the most used way to do to modify DNA, but that doesn't make it directly very easy or very cheap. Well, in general, it's cheaper than buying a hard disk or SSD for storage, but the use for it is so small. Currently they are going into more research and are going to see if they can find ways, if they can find unknown unknown processes within the bacteria that we don't know of that happen, or if there's something in there that we don't know the existence of yet. So first they are going to research the bacteria themselves better to see if what we know is exactly completely what we know. And then they are also going to develop this further, increase the possibilities, increase the quality, increase the space, and scale it up basically. Just scale it up until the point that they have the capabilities of reading memories from your brain with just a simple machine without actually causing a problem. Because if they have to look in your brain right now, they have an issue. They have to disturb the cells, they have to disturb you, and everything like that. But it would be much more easy if, for example, somebody has memory loss and they have to find the source. They can just take a look in the cells and be like, oh yes, it's this, and you just sit there, you set a machine against your head, and that's it. That would be amazing, incredibly amazing, but probably not very likely to happen within our lifetimes. So yes, thank you guys for watching once again. The sources will be down below as always. Subscribe if you didn't do that. Leave a like, leave a comment. Let me know what you think of this, because this is pretty bloody amazing that we are able to store DNA or use DNA as storage. It's not something they expected to happen very soon, but it happened, and it might be viable sometime. Maybe soon, maybe late. By the way, did I mention that the storage time of DNA is counted in thousands of years, unlike SSDs and hard drives, which are used in most computers? They count them up to 100 years and then your data is gone, but with DNA you go a factor of 10 up. So thank you guys for watching. Leave a like, leave a comment, subscribe if you didn't do that, and hit that goddamn bell button. And thank you for watching, and see you guys next time again.